Hello everyone. Today we are to discuss muscle tissues, and let's start directly from its classification, which is one of the most simple among all the tissues. So muscle tissue is divided into two groups, striated and smooth. The difference is very easy to understand. Striated muscles are striated. So if we make the longitudinal section through the fiber, we'll find out those dark and light regions alternating one with another. And this is what we call striations, and striations are absent in smooth muscle tissue. You see the section is homogeneous, no lines, so the tissue is smooth. Then striated tissue is further divided into skeletal and cardiac, and the difference between them is not only in location but also in the structure and function. And this is one of our primary purposes today, to understand not only their structural features but also the differences, to differentiate them, to distinguish them one from another, those three types. And also one more type is sometimes considered apart. This is the visceral group, and it is labeled with a green arrow because it has the same structure as skeletal, and usually those muscles are considered as skeletal, but they do not belong to the skeleton. For example, in the upper 1/3 of our esophagus, muscles, they have the same typical structures in our skeletal muscles. But of course, esophagus is not a part of the skeleton. Yeah, that's why please keep in mind that skeletal muscle tissue not always is in the skeleton. Okay, the muscle tissue. Yeah, genetical classification. Also, according to the embryonic development, muscle tissues are divided into some groups. If to tell you the truth, I think that there is no reason to remember those 5 types, because when we investigate every particular system of organs, we'll return to the embryonic development and investigate sources of muscle tissues in every particular system of organs. So now please remember just in general that muscle tissues, they derive from all 3 germ layers, being ectoderm, mesoderm, endoderm, and even from the neural tube, from the neural crest, from the planktopleura, from all the sources. So really the muscle tissue is ubiquitous. Okay, now let's investigate in general every out of those 3 groups. We start from skeletal muscles. They are striated, and first let's look at the skeletal muscle as a whole organ. The organ is covered with a connective tissue, with a loose connective tissue, as it brings vessels, nerves, and immunity to the muscle. And this thick layer of loose connective tissue is called epimysium. Epi like above, yeah, epimysium. Then the whole muscle is subdivided into bundles of fibers with a connective tissue which is more thin, and it is called perimysium. Perimysium, here it is shown in the specimen. And even every muscle tissue cell, even every skeletal muscle fiber, is surrounded with a thin layer of connective tissue which is called endomysium. So around every cell, soft connective tissue, loose connective tissue, forms a special microenvironment to provide the proper exchange of gases, metabolites, and innervation. Now let's talk about the structure of every particular cell in the skeletal muscle. So in skeletal muscles, some cells are very long, and because their diameter is much less than its length, cells are called fibers. And this is the subject of confusion, so do not confuse. Fibers are not the components of the extracellular matrix. In the skeletal muscles, fibers are the cells, so cells are called fibers. Then let's investigate their structures in details. Inside those fibers, so every muscle cell is called fiber, and let's investigate some another terms. The cytoplasm inside this fiber is called sarcoplasm. Then smooth endoplasmic reticulum, which is very well developed and it sequesters calcium, is called sarcoplasmic reticulum. The membrane is called sarcolemma. So the outer plasma membrane is called, instead of plasmalemma, sarcolemma. Also, in the sarcolemma, a special system of invaginations is present. here this system is. You can see that sarcolemma sends finger-like invaginations inside the cytoplasm, inside the sarcoplasm, and in this way the excitation of the membrane is spread to the deeply located myofibrils. As you can see, myofibrils, or contractile filaments, they are located not only under the membrane but also in the center of the cell. And to excite them all together, not only superficial but also deep myofibrils, special invaginations are needed. So once sarcolemma get excited, T-tubule system spreads this excitation into the deeply even located myofibrils, and they all get contracted simultaneously. It's very important. So let's summarize the information. Inside the skeletal muscle, cells are called fibers. Here this muscle fiber is shown. Inside every cell there are myofibrils, or contractile filaments, and in every myofibril, to understand its structure, we can subdivide a fragment which is called the sarcomere. So sarcomere is the morphofunctional unit of the myofibril, and here this fragment is given, and let's investigate it in details. So in the sarcomere, borders are designated by special Z lines. Here they are shown. These are the Z lines. Then in the middle of the sarcomere there is an M line, and M line is used to fix myosin, thick myosin filaments, which are red in this diagram, while Z line is used to fix actin filaments, which are thin and gray in this diagram. So this is the typical structure of the sarcomere, and let's investigate it in details now. So, as in the center of the sarcomere, myosin filaments are fixed to the M line, and myosin filaments are thick. This region looks like more dark, and even under the electron microscope we can see that the central region of the sarcomere is more darkly stained. Moreover, in the central part of this dark line there is a more lightly stained region, which is called H zone. What is the difference and why the central region is more light? Because at the periphery of this dark region, between myosin filaments, actin filaments are located. So at the periphery we have both filaments, while in the center we can see only myosin, only thick. So the central region looks like more light because only myosin present, while at the periphery it is more dark because both myosin and actin present. Now we have to say that this dark region is called A band. A because anisotropic. Anisotropic means that it contains not the same type of the filament, and iso means not equal, means it contains not only myosin but also actin. While in the alternating I band, you see between the dark regions, between the A bands, we can see I bands. In the I band only actin filaments are located. So this region is something like homogeneous. That's why it is called isotropic. Iso means equal. That's why here only actin filaments are located. Okay, so I bands, isotropic bands, alternate with A bands, which are anisotropic bands. And this is exactly the nature of striations. That's why in skeletal muscle tissues as well as in cardiac we have dark and light regions. In the dark regions myosin and actin are concentrated, while in light regions only actin. Okay, in the center of the light regions, in the center of the I band, Z lines are located. In the center of I band of dark regions, M line is located. So the fragment between the two Z lines is called the sarcomere. Okay. Then this is in general, and the information explained in this in text is given in a slide. And now let's investigate it in details, because this is the oversimplified structural version. In details, myosin or thick filaments are represented by heavy chains, which are shown here, and they are intertwisted with their tails. And in the head region, there are active sites to interact with the actin. When muscle is relaxed, actin and myosin are independent one with another. They do not attach. They are not attached. Okay. So also there are some light chains. They are not shown in this diagram. Notice that myosin filaments are considered as thick because they are made up of many such molecules collected in a bundle and fixed in the center by the M line. After that, what about the actin? Actin is made up of globular fragments which are called G-actin, like in the cytoskeleton. You remember this arrangement. And two intertwisted chains of G-actin makes an F-actin, or fibrillar actin. But actin filaments, they contain a lot of additional proteins, and you have to remember them. It is important to understand the nature or the mechanism of muscle contraction. So, actin usually is not in contact with the myosin because this contact is blocked by the green protein, which is put in the groove between the two actin chains. So this green protein is called tropomyosin. And it usually prevents myosin attachment to the actin when muscle is relaxed. So tropomyosin function to block the interaction between actin and myosin. After that, you can see on regularly repeated distances troponin molecules, which are the complex of three: troponin C, I, and T. Troponin function is to attach calcium, and once calcium get attached, then troponin removes tropomyosin from the surface of the actin, and it allows myosin attachment. So troponin function is to allow myosin attachment to the actin in case calcium is present. Okay, so when muscle is relaxed, there is no calcium, tropomyosin prevents attachment of the actin. Once calcium is present, it binds to the troponin. Troponin removes tropomyosin, then myosin get attached to the actin. This is the first event in the muscle contraction. Okay, so now two additional proteins you keep in mind: tropomyosin and troponin. Why troponin is so important? Because usually in case of myocardial infarction, it appears in the bloodstream, and detecting it in the bloodstream, we can confirm the myocardial infarction in the very early period. This is a very important test in practical medicine. Okay, then are you ready to get some more information about proteins? Okay, so a little bit more about actin. Globular actin, they form two intertwisted chains. That's what we have already discussed. Tropomyosin, the same information is in text. It blocks the interaction with the myosin. Then troponin is present on the surface, and it contains 3 subunits, and it is used as a calcium sensor. After that, one more protein is present. This is the tropomodulin. It is the capping protein, and it terminates the polymerization of actin. So it regulates the length of actin filament, which is important in the structure of the myofibril. After that, another protein is nebulin, and it is not shown here, as I guess, yeah, and it is used as a ruler and also helps to regulate the length of filament together with the tropomodulin. And then some additional proteins also are present. Among them, the most important are put in this slide. First, titin. Titin is like a spring. It fixes Z lines to M lines and to the myosin, and its function to prevent overstretching of the sarcomere. Yeah, it prevents the maximal stretching, and also it helps to center the filaments, the thick filaments between the two thin filaments. Then desmin. Desmin, it is used in the Z lines, and it is used, as you can see, to fix the Z line to the membrane with the help of another protein which is called anchorin, like an anchor. So this means, please remember, this is the intermediate filament of the muscle tissues. And in case of tumors, if we don't know the source of metastasis, if we detect desmin there, we guess that the source is muscle tissue, that we should find for the primary source among muscles, like that. And I remind you that keratin is for epithelial tissues, vimentin for connective tissues, and neurofilaments and acid glial fibrillar protein for nerve tissue. This is from this topic, cytology, okay? So it has the practical significance. M line, it also contains some other proteins like M protein, creatine phosphatase, and myosin. Just remember that. And dystrophin is a very important protein. Here it is shown. As you can see, it link laminin with the actin filaments. Laminin, as you remember, this is the part of the basal amino. And basal amino is present not only in epithelial tissues, but as well in muscles. Around muscle fiber, there is something like basal amino. And exactly dystrophin, with the help of transmembrane proteins, which are the dystroglycan and sarcoglycan, dystrophin helps to fix actin to the laminin. And mutations of this protein results in the muscular dystrophy, Duchenne muscular dystrophy. And this is the genetic pathology, unfortunately widespread. That's why you have to remember. And the questions about dystrophin and about Duchenne muscular dystrophy could be found in the USMLE database. That's why please remember. We ask you to remember about these proteins, not because it is interesting to know, but it has the practical significance, really. Okay, let's move on. And now, when we know everything about the structure, we have to talk how it works. So, how this mechanism is realized. First of all, we have to say that in case muscle is relaxed, myosin is not in contact with the actin. As you remember, tropomyosin prevents that. So what happens? When muscle contracts, it is very important that the total length of myosin and actin remains the same. So what is changed is the length of the sarcomere, but not of the contractile filaments. And that's what we call sliding filament theory, when actin just slides on the surface of the myosin and the total length of sarcomere decreases. So actin slides along the myosin. And how this mechanism is performed, we'll talk a little later. But now let's look how the appearance of the sarcomere changes along with this process. As you can see, this is the structure we have discussed. Between the two Z lines, there is the sarcomere, there is a dark A band and light I band. What happens when actin get involved inside the A band more deeply? Along with the contraction, myosin pulls the actin, and actin moves to the center, to the M line, even being overlapped in the center. As a result, you can compare the length of this sarcomere with the length of this one. Of course, this length is shorter. So myofibril decreases its length along with the process of contraction, and the muscle as an organ becomes shorter, of course. So how it is reflected in its appearance? As you can understand, the H zone disappears because now actin filaments are deeply inside the A band. And as well, I band disappears because actin from the region of the I band are now totally inside the A band. Okay, I think there is nothing to... Looking at this image, it's very pretty clear. So how myosin pulls the actin to the center of the sarcomere? Let's investigate this mechanism. First, what we have to say that there are two molecules very important in this process, calcium and ATP. Calcium is needed to attach to the actin. As we have discussed many times, when calcium is absent, myosin and actin are not in contact. Once calcium appears, myosin attaches to the actin, and next step is the binding of ATP molecule. What is the function of ATP? There are two very important functions. First, ATP helps myosin to detach from the actin, and the second important step is ATP helps to unbend the myosin. So from bend position it unbends. Why it is important? Because after that it attaches to the next site of the actin, and now ATP is hydrolyzed into the ADP and phosphate. Yeah, and once phosphate group detaches, myosin bends, and this is exactly what helps to pull the actin to the center of the sarcomere. And after that, when ADP detaches, new molecule of ATP appears, yeah, and it helps to detach myosin and to unbend it. Once it gets attached to the next site of the actin, ATP is hydrolyzed, so it bends. Then new molecule of ATP comes, it unbends, it detaches, unbends, and the regular repeat of this cycle helps many times to pull the actin, like to pull the rope to the center of the sarcomere. So this is the cyclic event, because the only one action, only one cycle is not enough to pull all the actin filaments to the center. Many cycles are needed, many ATP molecules. So please remember that attachment is mediated by calcium, detachment is ATP molecule, and hydrolysis of ATP is needed to generate this pulling force. Okay, so calcium to bind, ATP to detach and to pull. And what is important in case of death, in dead body, in dead muscle, actin and myosin are strongly connected one with another. Now you understand why, because calcium is present, it couldn't be pumped to the sarcoplasmic reticulum, and because there is no ATP to detach actin from myosin. So in dead tissues, in dead muscles, the overcontraction is observed. So next, factors affecting contraction of the smooth muscle. Let's investigate the sequence of events. First, what happens? Nerve impulse excites the sarcolemma. It gets depolarized. Then, through the system of T-tubules, excitation is conveyed even to the deeply located myofibril. After that, as the T-tubule is in contact with the sarcoplasmic reticulum, calcium is liberated out of there. So excitation of the sarcoplasm T-tubules result in the influx of calcium inside the sarcoplasm. Besides, notice one more term you have to keep in mind. One T-tubule with the two closely attached sarcoplasmic cisterns are called triads. So the combination of the terminal cistern, T-tubule, and another terminal cistern is what we call triad. Okay, so excitation of the T-tubule results in the calcium liberation, and then calcium interacts with the troponin, myosin attachment takes place. Then ATP is needed to detach the myosin, to unbend it, and after the attachment to generate this pulling force by hydrolysis of the ATP molecule. That's what you have to know, but there are a little bit more details. There are some of them. So let's investigate several other proteins. If you want to get the Mark 5, you have to know them. So the plasma membrane of the skeletal muscle is equipped with a special ryanodine receptors. Here, sorry, I've confused. The plasma membrane of the sarcoplasmic reticulum is equipped with the ryanodine receptors, and by opening them, calcium get released to the cytoplasm. Then inside the sarcoplasmic reticulum there is a calsequestrine. This protein sequesters calcium, it absorbs calcium, and it helps to hold calcium inside the sarcoplasmic reticulum, and it is liberated only in case of contraction. Then in the T-tubules, in the sarcolemma of the T-tubules, we have dihydropyridine sensitive receptors, And exactly those receptors, by excitation, they are also used at the calcium channels, but their main function is to affect ryanodine receptors, making them opened. So excitation of the sarcolemma results in the excitation of the change in the conformation. It changes the conformation of dihydropyridine sensitive receptors in the T tubules. After that, ryanodine receptors in the sarcoplasmic reticulum get opened and calcium is released to the cytoplasm. Besides, what is interesting in cardiac muscle tissue, dihydropyridine sensitive receptors also are used as calcium channels. So in cardiac muscle tissue, calcium enters through these receptors, and after that additional portion of calcium is liberated from the sarcoplasmic reticulum. So in cardiac muscle contraction, this period is a little bit longer and the cycle of contraction is longer. But in skeletal muscles, only sarcoplasmic reticulum is the source of calcium. Okay, it is important in physiology. Okay, let's move on. And now we talk about 3 different biochemical differences between different muscles. In sports, there are 2 strategies like marathon runners and sprinters. And 2 types differ significantly one from another. So very successful marathon runners are very bad sprinters and vice versa. So the opposite is true. And why? Because they have different structural features of their muscles. And in marathon runners, red muscle fibers prevail, while in sprinters, white muscle fibers. And what's the difference? What is the nature of those strategies? The differences in the biochemistry. First, if we talk about red fibers, they are especially numerous in marathon runners. In people running for a long distance, but not very quick, but for a long time. So the preferences is not the speed, but the distance. For a long distance, for a long time. In these fibers, they have special molecules called myoglobin, and it is very similar with hemoglobin, as it helps to capture oxygen, so they have their own oxygen storage inside them. Why? Because they oxidize glucose with the help of oxygen. And you remember that aerobic oxidation gives 38 ATP molecules out of 1 glucose. So it takes some time, but it gives us 38 ATP from 1 glucose, but for a long period of time. So these fibers, they are red because of myoglobin, and they need this oxygen because they get ATP by oxidizing the glucose. And they are slowly working, relatively slow working, but they work for a very long period of time. And they slow twitch, fatigue resistant. For example, in our back, some muscles, they help to maintain the posture, and they are always contracted, but they work slowly, but for a very long period of time without fatigue. And of course, these muscles, red muscles, prevail in the marathon runners. Then another type, opposite type for sprinters, is a white type. They do not oxidize glucose, but they prefer to use anaerobic glycolytic processes. Anaerobic glycolysis gives us only 2 ATP out of 1 glucose, but very quickly. And their main strategy to work very quickly, but for a very short period of time. They get tired very quickly. And that's everything because of biochemistry. So they don't need myoglobin, they don't need oxygen. They do everything without oxygen. They need many glycogen, many glucose molecules. That's why they have internal glycogen storage. And they have fewer mitochondria, less myoglobin, of course. So they are fatigue prone. Of course, they get tired very quickly, but they're very fast. And they are abundant in short distance sprinters, weightlifters, and so on. There are also intermediate type. It is called type 2A, and it has something like equal mixture of both. And this is the intermediate strategy, of course. But it's very easy to remember if you connect and associate the strategy, oxygen, and the structure. Let's move on. So let's summarize skeletal muscles, and as after that we go to discuss cardiac muscle. Let's fix at the structural features of skeletal muscle tissue. So first of all, in skeletal muscles, cells are multinucleated. So skeletal muscle fibers, they contain many nuclei in the one cell. After that, those nuclei are at the periphery. And to understand the position of the nucleus, always look at the cross section. If you are given only longitudinal section, it's very difficult to understand whether nuclei are in the center or at the periphery. Only cross section gives you clear vision that nuclei are at the periphery. After that, triads are present. So this is the combination of T-tubules and adjacent cisterns of sarcoplasmic reticulum. That's what we have discussed. Then T-tubules are located on the border between the I and A band. Between the dark and light regions, between them, every T-tubule is located. And then there can voluntary contraction. So if we can control the muscle, then obviously it is voluntary, it is skeletal. And this is the feature of skeletal muscle, because cardiac and smooth muscles are not under the consciousness control. And of course, another important aspect is the regeneration. Skeletal muscles, they do regenerate up to 20%, could be regenerated in case of young organism. And the source of regeneration, these are the satellite cells. They are located at the periphery under the plasma basal lamina. And POX7 is the marker of those cells. In immunohistochemistry, it could be revealed with the help of antibodies, anti-POX7 antibodies. Also, muscle cells could be regenerated from the perivascular stem cells or pericytes, and also they are the source of smooth muscle cells. Okay, in embryonic development, satellite cells are formed from myoblasts. So myoblasts are the primary sources of skeletal muscle fibers. By their fusion, skeletal muscle fiber is formed, and some cells are left in form of satellite cells. So the future regeneration results not in the formation of new fibers, okay, but by adding satellite cells to the pre-existent fiber. So fibers are regenerated by fusion with new cells. Then, now as for the cardiac muscle and its features, in cardiac muscle tissues, cells do not fuse completely one with another. That's why only one or two nuclei could be found in the one cell. But between the cells, borders are more more densely stained, and they are called intercalated discs. And this is the structural feature helping us to distinguish cardiac muscle from skeletal on the longitudinal section. So if you look at the section and on the background of striations, you can see those dark lines perpendicular to the long axis of the cell. This is the feature of the cardiac muscle tissue, for sure. Then, another structural features of the cardiac muscle. As you can see, the nuclei are at the center of the cell. In skeletal muscles, they are at the periphery. Then, there are no triads, as you can see that the T-tubule is in contact with the sarcoplasmic reticulum, but sarcoplasmic reticulum is much less developed comparing with the skeletal tissue. So here we can see T-tubule, there are no triads. Sometimes dyads are present, but very rarely. Then T-tubules are located at the level of the Z line. Remember, in the skeletal muscle, it is between A and I band. Here, it is at the Z line level. Then involuntary contraction, of course. Then myofibrils and cells anastomose one with another to synchronize their contraction, because in skeletal muscles, fibers contract independent. That's why we can perform very exact and a very scrupulous movement. In cardiac muscle, all fibers together. and presence of intercalated discs, as we have said, and also we have to say that in muscle tissue, in cardiac muscle tissue, a special source of development, it derives from the splanchnopleura, or the visceral layer of the lateral plate mesoderm. That's what's the difference. Then, what about the intercalated discs? So if we enlarge those junctional complexes, we'll find the combination of three. First, gap junctions. As you remember, they communicate to neighboring cells with ion channels, and in this way cells get synchronized one with another, which is very important for cardiac muscle. So if they are blocked or downregulated, then the contraction is not effective. Desmosomes, exactly because of desmosomes, those regions are so dense. And as you remember from the topic epithelial tissues, we considered intercellular junctions. They provide the mechanical coupling of cells. This is the most firm, firm contact to prevent tearing of the cell along with the contraction. And adherence junctions, those help to fix myofibrils to the plasma membrane, and that's how it works. Regeneration of the myocardium. Besides, in myocardiums, the process of contraction is almost the same as in the skeletal. That's why we do not consider it a part. And as I told you, those dihydropyridine sensitive receptors, they also are used as the calcium channels. That's the only one difference, I guess. Then, as for the regeneration of myocardium, this is a very important subject, a very important question of the modern medicine, because after myocardial infarction, the primary purpose is to regenerate cardiomyocytes and endothelial cells. There were lots of investigations, and it was thought for a long time that in the myocardium undifferentiated cells reside, and after the stress, after injury, they could be used at the source to regenerate cardiomyocytes. Actually, that's not true, and nowadays, according to the most precise experiments with the genetically modified mice, it was revealed that cardiomyocytes, even after infarction in regeneration, they are formed only from the pre-existent cardiomyocytes by division. And it is very important to understand that there is no reason to look for undifferentiated cells or to stimulate them to differentiate into cardiomyocytes. There is no such natural mechanisms helping to obtain cardiomyocyte from any external source. Only we have to stimulate existent those cardiomyocytes which exist to divide. And these are the two hyperlinks, very interesting articles. If you want to be sure, then please read. Also, I want to say that in regenerative medicine nowadays, even in humans, mesenchymal stromal cells are used. They are injected in the region of myocardial infarction or to the bloodstream. And it is believed that mesenchymal stromal cells, they differentiate into cardiomyocytes. But that's not true. And it was shown that their positive effects is only in producing biologically active substances. And those mesenchymal stromal cells, they only stimulate local cardiomyocytes to proliferate. But themselves, those mesenchymal stromal cells, they do not incorporate, or very low percent get incorporated. So that's what you have to know. And after that, let's talk about smooth muscle tissue and smooth muscle cells. They don't have striations, and according to evolutionary development, they are more primitive. They are something like more early developing myocytes, but they have their benefits. So one cell of the smooth muscle tissue is called smooth muscle cell. Okay, and its features is that it has thin actin and myosin filaments, but they do not form myofibrils. That's why there are no cross striations, because actin and myosin are not so well organized. Then nuclei are at the center, like in cardiac muscle. So calcium is pinocytosed from the outer environment, and this process is rather slow and it takes some time. That's why also the contraction is a little bit slower. Then T tubules are absent, and it is believed that caveolae used to form T-tubules in evolutionary development in future. So caveoli are like precursors for T-tubules in this case. And the contractions are involuntary in smooth muscles, we do not control them. After that, let's talk about the process of muscle contraction, because in smooth muscles it differs significantly from skeletal and cardiac. First of all, contraction is not always due to depolarization of the membrane. There are two ways to stimulate the contraction. First of all, to depolarize the membrane and to open voltage-gated calcium channels. That's what occurs in case of nerve impulse excitation or mechanical stretching. It helps to open the calcium-gated, voltage-gated calcium channels. Another way is when some hormones act on their receptors, for example, angiotensin or vasopressin. When they act on their receptors through the system of secondary messengers, they open calcium channels in the smooth endoplasmic reticulum, and in this way the concentration of calcium increases in the sarcoplasm. Okay, so two mechanisms, and sometimes the membrane is not depolarized, but secondary messenger system works. For example, oxytocin is another example, contracting smooth muscles in the uterus. Okay, then calcium interacts with a calmodulin complex, and calmodulin activates myosin light chain kinase, which in turn phosphorylates the myosin. Notice that there is no troponin in this system. Calcium interacts with the calmodulin, and myosin light chain kinase is needed to phosphorylate the myosin. As you can see, inactive myosin is in form of circle, it is coiled. When it gets phosphorylated, it uncoils and it fixes to the actin. And then ATP is needed to perform the cyclic contraction mechanism. What is important to understand in this mechanism? What is important difference from skeletal? Because here the contraction is very, very slow, but it lasts for a longer period of time. Because these are slowly contracting muscles, they hydrolyze only 10% of ATP comparing with the skeletal muscle for the same period of time. But it helps them to be more energy safe. This is like energy safe technology. They use less ATP to maintain contraction for the same period of time. And that's why this strategy is popular in our organism, internal organs. Yeah. Another reason why they contract for a long time is that just after phosphorylation, myosin attaches to the actin, and after this attachment, if it gets dephosphorylated, then it hydrolyzes ATP much less effectively, so it can't detach for a long time. And this contraction, this fixed contraction, is the mechanism which is used in our smooth muscle cells in our vessels, and it helps them to maintain the blood pressure. So contraction for a long period of time is maintained not because ATP is regularly used like in skeletal muscles, but because just after attachment myosin dephosphorylates and it decreases its ATPase activity. And you know that without hydrolyzing the ATP, myosin can't get detached. So this mechanism helps to maintain contraction for a long period of time. And why we highlight this mechanism? Because sometimes we need to decrease the blood pressure, we need to relax smooth muscle cells. And to understand how to relax them, we need to know how they contract. So nowadays the popular strategy is to decrease calcium level in smooth muscle cells, and those drugs are very widespread in pharmacology. And also the very prospective direction is to affect this mechanism of myosin dephosphorylation when it is attached to the actin. Okay, but this is very complicated, of course, but very important in understanding how to treat many diseases. So contraction of the smooth muscle cell. Now in text, it starts from the pinocytosis of calcium. After that, besides, instead of troponin, caldesmon and calpanin are used, and they prevent the contact between the actin and myosin in the smooth muscle cells. Then calcium calmodulin complex activates the myosin light chain kinase. That's what we have just discussed. And this kinase phosphorylates the myosin. As a result, myosin interacts with the actin. And then ATP is used to release this pulling force and to contract the cell. As a result of contraction, nucleus get coiled, and that's how we can confirm that muscle is contracted, by the shape of the nucleus. So, regeneration of smooth muscles, they regenerate very easy, and smooth muscle cells, they could be generated even from another types, from fibroblasts, for example myofibroblasts, they could give rise to smooth muscle cells. In epithelial tissue, some epithelial cells could form smooth muscle elements. Then smooth muscle cells themselves, they can divide even when they are differentiated, and at last the mesenchymal stromal cells could give rise to smooth muscle cells and pericytes of blood vessels. They are present around capillaries in some arterioles, and also by transdifferentiation they also can give rise to the smooth muscle tissue. So that's very important to understand how to regenerate this tissue. Relatively easy comparing with other types. Okay, I think this is the last slide of our lecture. Thank you for the attention, and goodbye.